I'm your host, Nick Jikomis, and today I'm speaking with Dr. Margaret McCarthy, a neuroscientist at the University of Maryland whose lab studies the origins and mechanisms of sex differences in the brain, the endocannabinoid system, and the neural basis of animal behavior. We discussed the nature of sex differences observed in the mammalian brain, including differences in the susceptibility to various diseases and disorders of the brain, differences in juvenile play behavior in males versus females, and how these can be altered by sex hormones and cannabinoids, sex differences in brain development that lead to sex differences in behavior, and how the endocannabinoid system in particular influences brain development and behavior. If you're interested in these subjects, this is a really fascinating episode. We get into why juvenile animals play and what the evolutionary purpose of that may be, and what the underlying brain development and brain mechanisms are that lead to behavior changes over the lifespan of organisms. As always, if you enjoy the content I'm putting out on this podcast, please like, share, and subscribe wherever you're listening or watching. You can find the video version on YouTube, and you can subscribe to my free weekly newsletter at mindandmatter.substack.com. On my Substack, you'll find all of the podcast episodes in both audio and video format, as well as some of the long-form writing that I occasionally put out that synthesizes what I'm talking about on various episodes of the podcast. You can also become a paid subscriber to support the podcast further, and I appreciate those of you that have been doing that recently. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it with the first meal of each day, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. And there's even evidence indicating that vitamin D deficiency is correlated with more severe cases of COVID-19 in those who get infected. Every time I go into the doctor each year for a checkup, I'm always told that vitamin D deficiency is very common and I should be supplementing on a daily basis. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase. And with that, here's my conversation with Dr. Margaret McCarthy. Dr. Margaret McCarthy, thank you for joining me.
Thanks for having me.
Can you start off by just telling everyone a little bit about who you are and what your lab studies?
Okay. As you said, I'm Margaret McCarthy, but everybody calls me Peg or Peggy if you're my mother. And what my lab studies is the developing brain with a particular emphasis on trying to understand how the brain develops differently in males versus females. And that's not, I just want to say that's just not because I'm trying to prove that, girls are smarter than boys or anything like that, but instead because of the enormous gender bias in neuropsychiatric and neurological disorders.
Yeah, no, on your lab website, your lab website is really interesting, and it's got some cool stuff on it, some facts and some diagrams and things. And you say that one of the aims of your lab, at least, is to understand the origins and mechanisms of sex differences in the brain. So as a neuroscientist and a biologist, what does biological sex actually refer to in the context of your research? And can you expand a little bit about why it's an important area of study?
Yeah. But it's also somewhat of a controversial area to study. So I want to just emphasize that I study the laboratory rat. So the laboratory rat does not have gender. It only has a sex. Gender is a purely human construct that involves one's self-perception and society's perception of one's sex. So in our rats, we only have males and females, and there's no gender fluidity. The reason it's important to understand it from biologically is that in humans, We know that clinically there's an enormous bias in what your relative risk is to be diagnosed with a developmental disorder, such as autism spectrum disorders, attention deficit and hyperactivity disorders, early onset schizophrenia, stuttering, dyslexia, Tourette's syndrome. Just about any early onset disorder is going to be more likely to occur in boys. It does occur, they absolutely occur in girls as well, but when we look at probabilities and relative risk, they're more likely to occur in boys. But if we flip around and look at things that occur predominantly after puberty, like depression, anxiety, compulsion, eating disorders, post-traumatic stress disorders, neuroinflammatory disorders, those are much more likely to occur in women, about twice as frequently as they do to men. So clearly there's some kind of a big a shift across the lifespan in your relative risk. But in humans, women, by the time that they become susceptible to these disorders, they've had decades of life living in a gendered world as a woman. And we can't separate out the influence of all of the cultural and societal expectations in that gendered world for both men and women to really get at what's biology versus what has been one's lived experience. So that's where the rats come in. It's because we can control all that. We don't have to put up with parents trying to raise them a particular way or dress them and speak to them in particular ways. We can just cut out all of that sort of experiential things and get purely at the biology of the origins of this relative risk across the lifespan.
Interesting. So there are differences in mammals, including humans, but also rats and other things, between males and females in terms of generally what you just said is in terms of their susceptibility to developing different psychiatric conditions.
Right. Now, of course, my rats don't have psychiatric conditions, right? They don't get autism, they don't get schizophrenia, et cetera. So what we do is we have to look at what we call sort of phenotypes that are consistent with those disorders in humans. So we look at anxiety-like behavior, say, or depressive-like behaviors. But there's, there's real limitations into what we can really model in the animals. What I tend to do more is I just try to understand the basic biology of the male versus female brain development, and not so much in terms of, and then I do also try to look at how it responds to perturbations. And what we find is that the basic developmental trajectory is fundamentally different in particular brain regions, not the entire brain, right? There's nothing I say is about the entire brain. It's really important to keep that clear, that there's just different regions. The amygdala develops differently than the hippocampus, which develops differently than the cerebellum, which develops differently than the hypothalamus. People tend to say, oh, a male brain versus a female brain. There's no such thing as a male brain or a female brain, right? Every brain is a mosaic, a combination of a whole bunch of different regions that work independently and intradependently, and which was why neuroscience is so fun and so complicated. I lost my train of thought. What question was I answering?
No, we're just talking about, the susceptibility to different conditions and behaviors and things.
Yeah. So I try to understand. So, for example, looking at the amygdala, which we know in humans is associated with a variety of social behaviors, fears, and things like that in adults, and anxiety. And it has been implicated in a number of neuropsychiatric disorders. And so I've just asked the simple question, does the amygdala develop differently in males versus females, and then try to understand, and how is that differently, and what are the consequences of that difference? Now, sometimes in other brain regions like the cerebellum, I will ask, how does the cerebellum respond to an injury early in life, and is that response different in males than females? So there's kind of two different ways to go about it.
Got it. So one of the things it says on the website, just to give people a little bit more here to anchor themselves, it says sexual differentiation in the brain is a developmental process whereby physiological and behavioral phenotypes are modified to match gonadal phenotype to ensure reproductive success. Can you sort of translate that for people that aren't familiar with all those terms?
Yeah, absolutely. So a lot of us are familiar with the idea that a male mammal is an XY chromosome complement and a female is XX chromosome complement. But the only consequence of that is that there's a single gene on the Y chromosome that codes for the formation of a testis, because there's a little collection of cells it's bipotential. It exists near the developing kidney, and it can become an ovary or it can become a testis. It's bipotential. And if there's a Y chromosome with this gene called SRY, which is sex determining region of the Y chromosome, that little group of cells will form a testis. And I always love to remind people that, we didn't discover this gene until the 1990s. Now, for many people, that seems like yesterday. For some people, they're like, well, I wasn't even born yet. Yeah, but for a lot of scientists, that's like yesterday. We only discovered this gene in the 1990s, a guy named Robin Lovell Badge in London. So if that gene is present, that little group of cells will become a testis. And if it's not present, that little group of cells will become an ovary. That happens stunningly early in development, right? In humans, it's within the first couple of weeks, right? And in the rodents, it's in the first couple of days. In rodents, then everybody thinks about the puberty as being the time when you first see steroid hormones, right? That's when testosterone comes, and that's when estrogen and progesterone come. But actually, in mammals, the fetal testis begins to make testosterone while he's still a fetus in utero. And in our rodents, that happens about 5 or 6 days before the animals are born. In humans, it happens in the second trimester. And there's huge amounts of testosterone that are produced, right? Very high levels, equivalent to adult circulating levels of steroid.
Wow.
And this is after the testis is formed and all of the reproductive tract is formed and everything. And so as far as we know, the only purpose of this surge, we call it a testosterone surge. I love the Japanese call it the testosterone shower. Near as we can tell, the only purpose of that is to act upon the developing brain, to sort of imprint on it, if you will, the physiology and behavior that is consistent with masculinization, masculine fitness. So in terms of the physiology, the brain controls the pituitary, and the pituitary controls the gonads, and there's a feedback loop. We call that an axis, right? The hypothalamic pituitary gonadal axis. And that axis functions very differently in males and females, because males need to make sperm all the time so that they're always sexually receptive, whereas females only ovulate periodically, in humans about once a month, and in our rodents about once a week. And so females in most animal species, humans being the notable exception, in most animal species, females will only mate right around the time that they ovulate. And that's the mating behavior is controlled by the brain. The ovulation is controlled by the brain. So if you're going to have those two things be in sync, you have to have the gonad and the brain in sync.
Right? I see.
And likewise, males have to be able to be ready to mate all the time for their fitness, right? So everything is about evolution maximizing the fitness of males and females relative to themselves.
And so I would imagine that, when we think about something like sexual dimorphism, the fact that in most sexually reproducing species, you have two morphs, that you have the female body plan and the male body plan, which correspond to different physical features, different physiological features of the body. When we think about sexual dimorphism, what are some of the ways that the brains of mammals tend to be sexually dimorphic that hold across species? Are there any general things that hold, or is it really like a species specific thing where, when we think about rats versus humans versus something else, it's going to be species specific?
Yeah. No, I would say that we have some generalities and some we haven't been able to ask the question yet. But probably so a lot of times, let me just reframe your question for an earlier question, because a lot of times people will just ask me, what are the sex differences in the brain? And that seems like that should be such an obvious question to answer, right? I should be able to say, oh, well, A, B, C, and D. But it's a more difficult question. So the brain can differ by the size of a particular region, right? So it could be bigger or smaller in males versus females. And that's what we call sort of a macro difference. In humans, it can be the amount of white matter versus the amount of gray matter. But then it can also be at the more micro level, it can be the number of synapses that are formed within a particular region, the number of axons that project from one region to another region, can vary enormously. The type of the neurons, right? They could be more inhibitory GABAergic or more excitatory glutamatergic, and the relative amount of each can vary. So, and then you could have the exact exact same cell population, but then the genes that they express can differ between males and females. So we could really go from big sort of macro, the hippocampus is bigger, down to the cellular, exactly what you express differs in males and females. Now, there are some generalities that we know about. One of the most famous is what's called the sexually dimorphic nucleus of the preoptic area, which was named for its discovery. It's a really fascinating kind of story, I think, a scientific story of how it was discovered. There had been attempts to look for sex differences in the brain in the 1960s. And if you just hold a brain in your hand, you can't tell a male from a female. They don't come in pink and blue, right? You literally do not know what it is. And two sets of scientists, a guy named Don Pfaff at Rockefeller, and then a pair named Raceman and Fields, they looked at very precise exacting, like using electron microscopy to look at, really, like they weren't looking at trees for the forest. They were looking at the bark on the trees, right? Really, 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 really detailed. And they found very small and subtle sex differences, one of them only in response to an injury, like they did a lesion. This was such huge news that it was published in Science. Both papers were published in Science because nobody had reported anything different in the male and female brain before. And then it sort of like nothing happened until the 1970s, when a guy named Art Arnold, who was also at Rockefeller as a graduate student, was thinking a lot about the brain, and he was thinking a lot about behavior, and he was studying birds, and he was thinking about canaries, in which the males sing this extremely complex, beautiful song. That's why everybody wants a male canary, and the females just tweet, right? They don't sing. And he speculated that had to be controlled by the brain. And so he took male and female canary brains and he sectioned them and just stained them to just, like we call it a Nissl stain, right? Just to visualize them. And he saw this huge nucleus in the males that was related to the song that was just tiny in the females. So it was available, you could see it with the unaided eye. It was such a big sex difference. So that was also a Science paper that he published along with Fernando Nottebohm. That stimulated a guy named Roger Gorski, who is at UCLA, who had been studying rat brains to understand that difference in the control of the gonads I was talking about earlier, the ovary versus the testis and ovulation versus spermatogenesis, to go back and look at his brains just from a distance. And then all of a sudden, this little tiny nucleus popped out at him that was much bigger in males and females. And it was at the macro level, right? So he named it the sexually dimorphic nucleus, and it happens to be located in the preoptic area, which the preoptic area has nothing to do with vision. It just happens to be a region that's pre to the optic chiasm in the brain. That was in the 1970s, and that really sort of stimulated just a huge amount of interest. And you could almost say, founded the field of sex differences in the brain. That nucleus exists in just about some variant of it in about every species that we've looked at, including humans. And there's an analog in humans that's considered the sexually dimorphic nucleus. It's called the anterior nucleus of the interstitial hypothalamus. And it was reported by Simon LeVay in the 1980s that it's larger in males and females, in heterosexual males and females. It was right around the times of the AIDS epidemic, when a bunch of brains were becoming available from homosexual men who died of AIDS. And that nucleus was smaller in homosexual men than heterosexual men. It was in between heterosexual women and men. It has since been discovered in sheep, in rams. A guy named Chuck Roselli has been studying them out west, in which there's about 10% of the rams are naturally preferring to mate with other rams, as opposed to mating with ewes. And in the sheep, again, the sexually dimorphic nucleus is different in size. And he finds that in the males that prefer to mate with other males, the nucleus is smaller and more on the size of the females. So that one really, really cuts across quite a few species. There are other examples as well, but some things, like a lot of the sex differences I've discovered are things like the number of synapses, right? Or the morphology of the astrocytes and things like that. And those are much harder to do in a human brain because, we don't have we can't do the kind of manipulations and microscopy and stuff in humans, or people just haven't done it.
I see. So you can get sexual dimorphisms in the brain, differences between males and females at sort of every level of resolution. Some of them are big enough that you can see them with the naked eye, and some of them go all the way down to the level of synapses and molecules and genes and things. Correct. And are there any general rules for when things are strongly dimorphic between the sexes? So for example, is it fair to say, or is it reasonable that most of the dimorphisms have something to do with brain regions that are related to reproductive behavior, or is it not limited to reproductive behavior?
Yeah. So that's an excellent question. And at the beginning of the field, the assumption was that they were only related to reproductive physiology and behavior. And in fact, that sort of relegated the field to sort of a sideline of neuroscience, right? It was considered sort of niche, right? And not particularly important. And then in the 1990s, a woman named Catherine Woolley, who at the time was a graduate student of Bruce McEwen, published a series of papers building on Maya Frankfurt and other people in the lab, showing that in the hippocampus, estradiol across the estrous cycle would change the number of synapses across the estrous cycle up to about 30%. And that was considered an absolutely crazy, heretical idea that could happen. In fact, they had to prove it many ways from Sunday, but she and Bruce McEwen are both outstanding scientists. And eventually the community had to accept that this was true. And this had nothing to do with reproduction, right? It had to do with learning and memory and with stress responding, which is what the hippocampus does. Now, that wasn't a sex difference, right? That was a hormonal modulation that she found. But that sort of broke open the idea that there could be sex differences in other regions of the brain that were related to other functions than reproduction. And many have been found. I still kind of tend towards thinking that they're more related to reproduction, but I'm often chastised and corrected on that by a colleague named Larry Cahill, who is absolutely adamant that the magnitude of the sex differences in the hippocampus and some cortical regions are just as great as those that are related to reproduction. And I will say that by and large, with the exception of the sexually dimorphic nucleus, which is really bigly different, it's about it's 3 to 5 times bigger in male rats than female rats. In humans, it's only about 1.5 times bigger in males versus females. Most sex differences are on the 1 to 2-fold level. And I think that that's by design. I think nature canalizes sex differences to keep the two sexes really separate, but not flying off in completely different directions, right? So you don't want them to be so divergent that it's, females are from Venus and males are from Mars kind of thing, because reproduction requires such delicate coordination. And plus also, a lot of times the two sexes are solving the same problem, right? Mm-hmm. Both sexes have to learn, right? Both sexes have to feel pain. Both sexes are susceptible to habit formation and hence addiction. Both sexes have to be stressed and et cetera. So they're not, most sex differences are not, huge, but they are what I would call reliable, right? They're usually not a continuum in my world. And the endpoints that I look at, you tend to have, you got the male and you got the female, and you don't have a lot in between.
I see. So if you, let's say that you just sort of picked a random chunk of brain in a random mammalian species, maybe it's a rat, maybe it's a human. If you sort of had infinite data, if you could go in and count all the synapses or look at the gene expression patterns, would you tend to find some kind of difference between males and females on average, or would that be exceptional, or would it, happen half the time?
That's a fascinating question. That would be a great experiment to do, because of course we don't do things randomly, right? We always have a reason. And I would say in my experience, I tend to look at a lot of fairly different regions. And for example, when we look at the cerebellum, we don't see any sex differences at all. It's developing just exactly the same until we perturb it. If we create hypoxic ischemic injury, or if we do a viral inflammation or a bacterial inflammation, then we see the sexes kind of fly apart. And that's sometimes called a latent sex difference or divergent sex difference. And that's also seen at the cellular and molecular level. But if I look anywhere in the hypothalamus, you'll find sex differences. Hippocampus, definitely. There's some cortical regions. I don't do cortex. I don't have a good answer to your question, except for to say that more often than not, if you keep digging, you'll find a sex difference. Now, one thing I have people come to me all the time with, I've got a sex difference. I found a sex difference. Oh my God, I'm so excited. What am I going to do? And I'll look at their data and I'll be like, yeah, I think you just have noise. I don't really, anything that's less than 1 fold is just a lot of times the sexes are not different. I don't feel like I'm giving you a really good answer because I don't have a clear answer. But I'm always telling my lab, they'll get all disappointed when something's not different. I'm like, it's just as exciting when it's not different. And it's just as important. And what we have to do, though, is we have to be as sure about no sex difference as we are about a sex difference. It has to be just as well powered, right? The ends have to be just as large and stuff. So like I have a whole new line of inquiry going on in my lab right now where we don't find a sex difference in one brain region in one response, but we do find it in another. And we're asking ourselves, wow, how are the two regions doing this so differently? And why? Again, why is it important that they not be different in one brain region and that they do be different in another brain region?
I see. So it sounds like it's fair to say that when you look for sex differences in the brain, if you look hard enough, you will often find them. But nonetheless, there are brain regions like the cerebellum where it appears to be more or less the same in both sexes.
Yeah. And there are probably many other regions where it is more or less the same. And there's always the reporting bias as well, right? Mm-hmm. So the reporting bias is towards finding a difference, and people don't tend to, when they don't find a difference, they don't report it, or it doesn't get attention, and et cetera. And of course, then I have a bias because I'm always looking for differences. So I might not be the best person to ask that. Other people might tell you, oh yeah, we never see sex differences.
And the other thing I want to go back to that I think is interesting is, so you mentioned a couple of things. You mentioned the hypothalamic pituitary gonadal axis. You mentioned this testosterone surge or testosterone shower, depending on your taste, that happens relatively early in development. With those things in mind, can you kind of connect the dots for people between the gonad, sex hormones, and brain differences that develop? Like, how does that start to happen, and how do those things all interact with each other to create some differences?
Great question. So I think you're right. I didn't complete that line of thought, which is, so that testosterone surge, what it is doing, it is getting into the brain. There are receptors in the brain for testosterone that they're not everywhere, right? They're only expressed in certain regions that are going to be responsive to testosterone. Testosterone is also a precursor to estrogen or estradiol. And there's a whole set of cells that have receptors for estradiol or for estrogen, and then those receptors are actually what we call nuclear transcription factors. So they actually directly interact with the DNA, and they cause the transcription, the turning on or turning off, often just as important, of particular genes. And then they also actually seem to mediate epigenetic changes, so that the turning on or turning off of particular genes is established relatively permanently, right, throughout life. One of the questions, so what I can do in my research program is I can take a newborn female rat and I can inject her with testosterone or estrogen, and I can watch experimentally the process of turning her brain into a male brain. And so it's a great experimental tool that I have. So I can monitor the process of sexual differentiation in real time, and I can measure various endpoints. Like I can look at the mRNA for various genes. I can look at the morphology of neurons, the morphology of astrocytes. I can look at the immune cell profile, et cetera, at the time. And that's how I get at the mechanism of sex differentiation. That all in my rodents is happening in the first couple of days of life. In humans, it's all happening in utero. So even if we could inject a human with testosterone, which we couldn't, we still wouldn't be able to study it because it's happening in utero. So even in primates, it's, like rhesus macaques and things, it's all happening in utero, which is we're fortunate that the postnatal rat remains sensitive to this. That's why we call it the sensitive period to testosterone. Now, if I take a newborn rat, I can do that up to about a week. Once she gets to be about 10 days old, if I try to inject her with testosterone, it's too late. She will be a female with a female brain for her whole life. And likewise for a male, if I take away testosterone really early, I don't let him get exposed to testosterone, I can make his brain female-like. But if he gets testosterone, once he's exposed, that's it. It's so the kind of the human analogy I give is There used to be a thing called a lazy eye. You had one eye that didn't quite look straight. And so the way that doctors used to treat that was that you would cover the good eye and you would make the lazy eye correct itself because it had to work harder. But if you didn't do that early in childhood, the eye would never realign. So there was a sensitive period during which it could happen, and then it's too late. And that's the nature of development, right? Development has things that if they don't happen at the right time, they'll never happen. The really fascinating question to me, though, is in the sex differentiation case, is that everything is happening in a pup, looks like a little pinky eraser, right? It's not capable of any kind of reproductive function. It can't even walk, right? It doesn't even have its eyes open. And in that little pinky pup, in that little tiny brain, you're coding for its ability as an adult to either be, mounting and emitting and ejaculating, to be aggressive, to be highly anxious, to prefer the odor of females, all of these things that are not going to occur for months, right? And so I'm really fascinated. It's like, how is that stored in the brain? How is that coded in the brain to go all that time and then it's not expressed until after puberty? So really, we don't know anything about that.
Interesting. So when people think about like sex hormones, testosterone, estrogen, and things like that, these are literally molecules that are produced in the body. They go through the bloodstream and they can get into the brain, and not only into the brain, but they physically go inside the cells and directly, in some cells at least, directly sort of control which genes are coming on and off and having their effect through that kind of mechanism.
Precisely. Yes. And there was just a beautiful paper published in Nature by a woman named Jessica Tolkien at Cold Spring Harbor, in which she really lays out the genes she uses very, very modern cutting edge techniques in 3 different brain regions and shows exactly what's getting turned on early, what's getting turned on in adulthood, et cetera. Yeah.
I see.
Steroids people don't appreciate how powerful steroids are. They tend to think of them like, other hormones, say like insulin or something, but they're a completely different class of compounds.
Interesting. And so these things go inside of cells, they control their development, with the caveat that it really depends on what phase of development you're in. If it's at the right time, so to speak, they can have a huge effect, and they might have little or no effect if it's too late and some window of opportunity has closed.
Correct. Yep, that's exactly right.
And then the final piece that was interesting there to me was that a lot of the tracks for these sex typical or sex specific behaviors are being laid down way before the behaviors ever express themselves. So somehow these changes happen and develop, and then they are sort of latent or stored in the brain only to sort of do what they're going to do later on.
Yeah, that's exactly right.
So one of the things that we'll probably end up talking about, because you've studied it specifically, has to do with juvenile play behavior in rats, which is sexually dimorphic, and we'll talk about that. But before we get to the rat stuff specifically, can you just talk about juvenile play behavior in mammals generally? How common is it, and what do we think the general biological function it serves as? Yeah.
So thanks for that question. So first, a little background on play. In that it's juvenile play behavior, as you said, and so it only occurs during a restricted period of time, and it's usually in our rodents. It's like just around weaning and until puberty. And once you get past puberty, interest in play goes away, and instead you're interested in mating and being territorial and etc. And it's a mystery still to this day what the purpose of play is. Somehow we all think intuitively it must be important, right? But nobody, and it's debated by psychologists, psychoanalysts, parents, teachers, and neuroscientists. And it used to be a very intense topic of investigation by neuroscientists back in the 1980s. Because back then the rat was the favored animal model for neuroscience. Then the transgenics, the power of the transgenics mouse swept in the 90s. And of course, that just, really pushed aside most of the research in rats because it was so powerful to have this ability to manipulate the genome. And it turns out mice are actually one of the very, very few species that do not engage in this very complex play behavior. So what do I mean by complex play behavior? When you put two rats together, so just look just like puppies or kittens, they will chase each other. They will, one will pounce on the other, and the other one will roll over onto its back to expose its ventrum. And then they'll jump up and they'll reverse, and the other one will chase the other one, and they'll do the same things. They'll stand up and they'll box with each other. And sometimes they just wrestle, they just roll around and wrestle. And we know it's rewarding. They like it. If you let them play in a place that they condition to it, and they want to come back to that place. There's no aggression. There's never any biting. It's when it's going well, it's reciprocal. If you get into a situation where one animal is always dominating the other and sort of pinning it, the play extinguishes, because the other one's like, you're not any fun. You don't let me pin you, kind of thing. And that's also true, in so many species that we know of, that we all, all of our pets and everything. One of the fascinating things I think about our pets is that they are, particularly dogs, they still play post-puberty, which is really another interesting question. The reason that I got so interested in it is that across all species that play, males will play more intensely, more physically, more frequently than females. Females do play. They are very physical. They're just not quite as intense and physical as males. And what fascinates me about that is that when they are at the age that they're playing, there's no steroid hormones around, right? This is like, think about like a 6 or 7 year old child, right? They don't have any hormones at this time, right? So the sex difference is coded by the hormones early in development, just like adult sex behavior and adult aggression, but it's expressed at a time that there's no hormones around. And so that means you're really comparing apples to apples, right? As opposed to in adulthood, when males and females have very different hormonal profiles. And so I also think that the fact that there's this difference between males and females can maybe open a window into what is the purpose of play. Do we really need play, kind of thing? And that's one of the avenues that my lab is exploring right now.
One way I suppose you could look at that, and I don't know if this has been done, has anyone simply tried depriving juvenile rats, say, of play behavior and see what the downstream consequences are for their reproductive behavior or something like that?
What an excellent idea. Yes. That was done. It has been done many times back in the 80s, but mostly it was done by just completely isolating the animals. So putting them alone in a cage away from all other animals. And it turns out that's about one of the meanest things you can do to a juvenile rat. They're very, very social animals. And it's very, very stressful for them to be isolated. So what we've been doing in my lab, and I should also say it was all only done in males, because all of the research back in the early days was only done in male animals. So one of the things that we've done, a graduate student named Ashley Marquardt in the laboratory, is she created a cage, a normal rat cage, in which she put a plexiglass perforated barrier down the middle, so that you could put an animal on each side and they can see each other, they can hear each other, they can smell each other, they can tiny little bit touch each other, but they can't play. And so she houses them like that through the entire juvenile period. And then she gets them through, takes them back out, puts them back into group housing at puberty. And then they go on to, and then adulthood, she's measured a whole lot of behaviors. And what she has found is that the males show impaired sexual behavior. They're slower to interact with the female sexually, and they interact less frequently. They show a longer latency. They have fewer mounts, fewer ejaculations, etc. Conversely, they seem to be hyper aggressive. So sometimes people say, oh, play is a rehearsal for aggression. No, play is probably a rehearsal for appropriate aggression, not, and so they're much quicker to attack a stranger, etc. They have a very bizarre response to sort of a social preference test. And it would say also it's like a hyper exaggerated response, which we think has to do with dominance hierarchies and not knowing how to interact to establish dominance hierarchies. And then they show a little bit of cognitive impairment. Fascinatingly, the females are completely normal. They show no effect whatsoever of having been deprived of play. And further, if in the middle of this period of this, not being allowed to play, if we free them from these barrier cages and allow them to have a play date, the males are exuberantly playful, right? I mean, they just show a huge exaggeration in their play. And the females are like they just are, again, unaffected. So it's a really fascinating difference. That why would the play be important to one sex and not the other? And again, it's just, we're just really fascinated by why is it the females, are they just resilient to the stress? When they sit in the cage together, I keep going like this because they sit right next to each other. They know the other animals across the barrier, right? And they sit right there. And somehow that's just enough socialization for the females. I don't know, maybe they're chatting away or something.
Well, yeah, I mean, that was another question I had, is when we talk about this play behavior in rats, we're referring typically to this easy to measure, easy to see, like rough and tumble wrestling type behavior. Conceivably, there's other ways they interact that are more difficult to discern.
Yes, that's an excellent point. And in fact, the play is so fast and vigorous, we have to videotape it and then slow it down to score it. And so one of the exciting things that's happening in neuroscience right now is computer vision learning. So you can use artificial intelligence and so we started this process. There's various programs out there. There's quite a few of them that are developing to see if the computer can see something that we're not seeing, right? But it's the challenge is that these two animals, you can teach the computer to track one, and we can get the computer to follow them around as they're like chasing each other, but as soon as then they interact and then they become a furry ball, right? And then it's really hard for the computer to tell who's who. So we're working on that, but you raise an excellent point, is that we do think that there could be things that are going on that we aren't detecting. There could be ultrasonic vocalizations that they're making to communicate with each other that we can't discern. And again, these are just technical problems because we can record ultrasonic vocalizations, but we can't tell which one is vocalizing, right? Because so things of that sort. So, but I think those are exciting questions to pursue in the future.
Yeah, well, you said a couple of things that were super interesting. So when we think about depriving the rats of play, there was this dimorphism there. So the deprivation of play affected males more than females. It resulted in subsequent deficits in their adult behavior, such as the reproductive behavior and their propensity towards aggression, as you mentioned, which is maybe a little bit counterintuitive to people, but I think not so much once you think about it. If you don't let the male rats do rough and tumble play with each other, then they become hyper aggressive. They basically don't know how to appropriately interact with other males and other members of the species later on. And you also said that if you deprive them for a little while, then they become like really, really excited to play even more. So that implies that this is almost homeostatically regulated the same way that other processes in the body are, which would also say that something very crucial is happening here.
Yeah, that's interesting. I'm glad you said that. I hadn't thought about it in terms of being homeostatically regulated, but you're absolutely right. In fact, we know sometimes in our experiments, if we want to do things like we want to look at the neurons that are activated by a play bout, right? So we'll look at an immediate early genes expression in a particular brain region after a play date. What we'll do is we'll isolate the animals for 24 hours before the play date. And then we know we'll get this huge exaggeration in play. And so you're right. It's like, or other times what we do is we let them play together every day for like 10 days in a row. They meet a playmate in the afternoon that they go, and it's not their cage mate, it's not their sibling, it's their playmate. And they go to a neutral arena, it's not in their home cage, and they get about 15 minutes to play. And they'll just steadily play the same amount pretty much across the whole 10 days. Kind of thing. So unless we isolate them, and then boom. And if we introduce a new playmate, that it doesn't affect the males, but the females, they're like, I don't know who you are, and they don't play with the introduced animal.
And now, will juvenile male rats preferentially play with other males, or do they play differently with juvenile males versus females?
Yeah, that's a great question. If we look in terms of the intensity of play, the highest is male to male, medium is male to female, and lowest is female to female. And when you do mixed groups, because what we do now is we started out, we would do like 6 animals together, and we would just watch the all 6 animals, which was crazy. Yeah, because you got to label them and all that kind of stuff. It's a good question whether or not the males initiated more with other males. And that's where, like, if we could get really sophisticated, computer learning would be vision learning would be a good question to ask. But definitely, one of the challenges of studying the behavior is that it does require reciprocity. And so if the other animal is not playing the same, that will kind of extinguish it. And so we've been looking at things like rats with genetic mutations for genes that are identified in autism, and to see if their play behavior has changed. And it is, and it's reduced. And what we have to do is we have to put the two, like the genetically mutated rats two together. Like, we can't do a mixed dyad because it won't reveal the effects, but so you have to sort of exaggerate it. But anyway, I whether or not there's a preference for a male with a male, I don't think so, but they'll have more reciprocity from a male. And so that just naturally exaggerates the behavior.
I see. So this play behavior is sexually dimorphic, both in terms of how vigorously and how frequently it happens in males versus females. It's also dimorphic in terms of how important or influential it is on their subsequent adult behavior. Because if you deprive males, they have deficits in reproduction and aggressive behavior, as you said. You don't see that in females. So the next question is really, okay, this is super interesting. Where does it come from? And I know that a lot of your work has looked at how this type of difference develops and how the endocannabinoid system in particular ties into that. So can you start to tell us that story of where this dimorphism comes from?
Yeah. So first you have to start is, what is the neural circuitry of play, right? In the brain in general, where does play come from? And there is a very strong consensus in the field now of what we call the social behavior network. And now there's also what's called a core aggression network. And these are brain regions that are interconnected that regulate social behaviors, and then also aggressive behavior. And they usually start with the olfactory system, either the main or accessory olfactory system. And then you're going to have projections to the amygdala. You're going to have participation of the prefrontal cortex. You're going to have a component of the reward pathway, the VTA, nucleus accumbens, the BNST, and then you sort of kind of go down to the midbrain to the spinal cord. The neural circuitry of play, near as we can tell, we don't know it as well as we do the others, because a lot of that has been really finely mapped now in the mouse. The neural circuitry of play is overlapping with the social behavior network. It's the same network, which is also fascinating, because then, like, why does play go away, right? Why does it extinguish with puberty? But fascinatingly, the sex difference is only in the medial amygdala. You could imagine it being a distributed property, right? That would it would be redundant through the entire social behavior network, but it's not. It's only in the medial amygdala. And why do we know that? We know that because if you take a newborn female rat pup and you put testosterone only in her medial amygdala, 3 weeks later or 4 weeks later, when she starts to play, she'll play like a male. Right? You don't have to have the testosterone anywhere else in the brain. So
I see.
We know that.
So there's one spot, medial amygdala in this case, you put testosterone in a female, so it's at levels that resemble what would be naturally in a male. And later on, when she becomes a juvenile, she'll play like a male.
Right. So my question was, what is that testosterone doing, right? What's its cellular mechanism of action? And we had been doing some other studies on the hippocampus where we had seen some sex differences in neurogenesis, actually with males making more new neurons than females in the first couple of days of life. And I had a new postdoc in the lab named Desiree Krebs-Kraft. And I said, well, the amygdala is in the same sections as the hippocampus. Why don't you just go and look at the newborn cells in the amygdala and see if there's any sex difference there? So she did. We do this with a BRDU injection. That's a thymidine analog that labels newborn cells. You have to then section the brain and look at it. And she went and counted the newborn cells in the medial amygdala. And lo and behold, she found that there were more newborn cells in the female than in the male. And that was exciting because, usually everything's always more in the male, more in the male, more in the male. And so she's like, oh, well, that's cool. So there's more newborn cells in the female than the male. And then she's like, well, now what? I was like, yeah, now what? That's a good question. Well, sometimes science occurs in funny ways. I had a colleague named Brad Alger who studied the endocannabinoid system. He was one of the pioneers of the system. And after, at this point, I think we've been colleagues for I don't know, 20 years or something. And every single seminar, every thesis defense, every journal club, every thesis proposal during the question and answer session, he would stand up and say, endocannabinoids can explain that, whatever it was, whatever phenomena it was, literally. So I just literally said to her, I don't know, let's look at endocannabinoids. Maybe they're regulating that's literally how it happened. And so she said, okay. And she took a pan agonist for the CB1 and CB2 receptor, which are the two receptors on which endocannabinoids and also THC act. And she injected animals. We just did a pharmacology experiment, and she was able to reverse the sex difference. That she was able to bring down the number of newborn cells in the female to the level of the male. So she was able to eliminate the sex difference. And since we knew from the previous work in the 1980s of Bruce McEwen and Michael Meany that the medial amygdala was critical to the sex difference and play behavior, she then looked at social play behavior, and she found that it also, the treatment with the pharmaceutical drugs that mimicked THC had also sex reversed the play behavior. And that set us off on this journey to understand really precisely what the endocannabinoids were doing.
So just to reiterate, so you give testosterone to a female, specifically in the medial amygdala. Her subsequent play as a juvenile will look like a male. And the same thing happens if you give an activator of CB1, CB2?
Correct. And you don't even have to give it into the amygdala. We just give it under the skin. We give it sub-Q.
I see. Yeah. And does that happen also with THC itself?
Yes, it does. We haven't published that work yet. So yes, but we have, and this is Jonathan Van Rissen, who has been carrying on this work, and he's preparing the manuscript now, in which if we inject newborn pups with THC at exactly the same time that we would do testosterone, it has the same effect on the newborn cells, and it has the same effect on the play behavior. Interestingly, if he gives THC to pregnant rats earlier, so we're looking at, because we're trying to model human cannabis use during pregnancy, if we give it much earlier, it has the opposite effect in terms of play behavior. It actually suppresses play behavior. So it promotes play behavior in the young animals when you give it to the newborns, and it suppresses it during fetal, if it's given during fetal life. And we don't know the mechanism of that. But again, it's just the importance of timing, development. Mm-hmm. It's all about timing.
Yeah, you can get the opposite effect at two different times with the same treatment.
Right.
So is there some kind of interaction you guys are hunting down between the sex hormones like testosterone and the endocannabinoid receptors? Do we know exactly where that link is?
Right. So this is where the story got really, really interesting. We did all those obvious things, right? So we looked for sex differences in the endocannabinoid receptors and sex differences in the endocannabinoids themselves. And we did find that, in fact, males have a higher level of the endogenous endocannabinoid called 2AG. And endocannabinoids are a funny signaling system, right? They are a membrane-derived signaling molecule that has that's sort of continuously produced all the time. And so there's like a tone, right? You have like a certain tone of endocannabinoids that you sit at. And so the conclusion was that males have a higher endocannabinoid tone than females. And if we give females testosterone, they make more, they have a higher tone. It's either because they're making more or they're degrading it less. A big part of the endocannabinoid system is, it's like, think about it as how to think about it. But basically, you make it and you degrade it, and you can either make it faster or degrade it faster, and that will shift the amount either way. So it's definitely the endocannabinoid tone, not the receptors, that was different. But then it was like, well, what are the endocannabinoids actually doing? And we were thinking about the adult and all the things that are known in the adult about how it regulates synaptic physiology. They have, presynaptic inhibition, depolarization, induced suppression, et cetera. And none of that applied to the development of the amygdala, which it makes sense in retrospect because it's just not developed yet. It doesn't have all that synaptic activity. And so when we're trying to think about cell genesis, so the big question was, who are those newborn cells, right? There's born. And of course, you think it's going to be neurons. And certainly in the hippocampus, we had shown it was neurons. But again, in retrospect, it makes sense. They weren't neurons at all. It was the glial cells known as the astrocytes, right? So these are the sort of, had always been considered the support cells of the brain. But we now know they're actually very, very active cells, and that they're really important to synaptic physiology and et cetera. And Desiree first identified that in fact that it did look like it was the astrocytes, that there was more in the females than the males, Desiree Krebs-Kraft. And then Jonathan Van Risen really confirmed that, but he added in the really interesting and unexpected twist of that he brought in a third cell type called microglia. Now, the problem with microglia is their name. They are not glia. They are not astrocytes in any way. And they're not micro either, because they're 10% of your brain. What they actually are a modified immune cell. And it's a modified macrophage. And they are born very early in development, and they get into your brain and they take up residence and they stay there your whole life. And they're scattered throughout, they're peppered. Actually, we call it, they're tiled through your entire brain. Your entire brain is full of microglia, and they're there as your brain's immune system. They're unique to the brain. They're nowhere else in the body. And what Jonathan found was that rather than, it used to be, we thought microglia, we called them quiescent. We thought they just sat there waiting for an accident to happen. And then they would maybe, release cytokines or prostaglandins and respond to the injury. With the advent of the transgenic mice and the amazing things that you can do with transgenic mice, we were able to visualize them in the living brain and found out that they're not quiescent at all. They're actually moving around all the time, and they're always checking on the other cells around them. But what Jonathan found, and again, just to repeat, Jonathan Van Risen, is that these macrophages, these immune cells, were in fact engulfing the newborn cells in large phagocytic cups, the living newborn cells, and they were actually then killing and consuming them. Basically, by consuming, I mean they degrade them.
So these are effectively like immune cells of the brain. And just like a white blood cell or something in your body might eat a bacteria from an infection, they're eating newborn neurons in this case.
Newborn astrocytes.
Newborn astrocytes. Okay.
Yeah. Yes. That's exactly right. And in an exquisitely tightly controlled manner, which is what's also fascinating. And that control is coming from the endocannabinoid tone, right? So the higher endocannabinoid tone in the males acts on the microglia to make them basically give them the munchies. It makes them hungrier, and they consume more of the newborn astrocytes. And so the females wind up with more of those astrocytes surviving. And so that as they get to the time of the juvenile playtime, the female's medial amygdala has more resident astrocytes than the male does. And you still say, well, what does that have to do with play? Well, what Jonathan Van Risen is showing now is that those astrocytes are actually inhibiting the play neurons because they're releasing adenosine, a chemical called adenosine. So the more astrocytes you have, the more inhibition you have of the play neurons. And the males have escaped from that break, basically, by killing off their astrocytes when they were young.
I see. I don't know if I want to go on this tangent, but you said adenosine is involved here. That immediately makes me think about caffeine.
Oh. Wow. Yeah. I don't know.
Okay. So so basically development is happening in males and females. Naturally, under normal conditions, you'll have different levels of endocannabinoids. They'll be higher in males, I think is what I'm hearing. And that will make the microglia hungrier, and they'll literally eat and digest these things called astrocytes. And that sort of releases the ability to have more play behavior in juvenile males and suppresses it in females.
Exactly. And interestingly, they only eat the newborn astrocytes, right? They like the babies, right? Which is, once the astrocytes are mature, they don't seem to consume them.
So a natural question here is, similar to what I asked before about deprivation of play. You said that if you deprive a male of its ability to do rough and tumble play with other juveniles, it has downstream consequences, negative consequences on the adult behavior, both on the aggression side and the reproduction side. What happens to adult behavior if you say masculinize the females or do the reverse? Does something similar happen downstream?
Yeah, that's an excellent question, and we have not done that experiment. That's because and I think that, so if we gave hormones to do that experiment, it like to peripherally say, then it would masculinize everything, right? Because it would invoke another mechanism in the hypothalamus and another mechanism in the hippocampus, etc. If we give the endocannabinoids, it's interesting. What we have to do, so when I give a newborn female testosterone and I turn her brain male, and if I take her to adulthood and I want to see her do male sex behavior, I have to give her testosterone again, right? Because even a normal male will not show sex behavior without testosterone.
I see.
But you raised an interesting idea. I have not thought about if we gave the endocannabinoids, could we get them with just adult testosterone to start to show male-like sex behavior? That's an interesting idea. I got to think about that.
One potential prediction would be that, if you're masculinizing the play behavior and the juvenile play behavior is causally related to adult reproductive behavior, at least in the presence of testosterone, would a masculinized female start exhibiting male-like mating behavior as an adult?
Great idea. I like that. I think we'll try it.
Interesting. What else can you say about this general subject? Is there anything that we've missed in terms of what your lab has studied with respect to the endocannabinoid regulation of this particular dimorphic behavior, or did we kind of cover most of the story there?
I think we covered most of it. Yeah, we're now trying to hunt down, further details on exactly which cells have the receptors and who's making the endocannabinoids and things. But yeah.
And another thing worth talking about. So you mentioned that, if you give some of these things, whether it's hormones or endocannabinoids, at different phases of development, it can have a very different effect. It can even have the opposite effect depending on when you give these things. So can you talk a little bit more about sensitive periods and like what is determining that? Why is it that giving, a molecule of some kind, whether it's testosterone or an endocannabinoid or THC or whatever, what's actually allowing it to have two different effects if you give it relatively early versus relatively late?
Yeah. Another really fascinating question. Why does what makes the sensitive period end? Right. Would be another way of putting it. And that's a really interesting. We know that we operationally define the onset of the sensitive period as the time when male fetal testes start to make testosterone. But why do females eventually become insensitive? Why can't I just give a female at 30 days old and change her brain into male? And it used to be because we thought, well, the events that are changing are things like cell death, right? And cell proliferation and the wiring up of synapses and things like that. And those are all considered permanent endpoints, right? You can't undie. And, you can only be reborn. And then we think of synapses, synaptic wiring is like, plugging an extension cord into the wall, right? And there I've made my synaptic connection and there it will stay forever. But interestingly, we now know all of those things are much more plastic than we thought. Synapses come and go, and actually cells continue to proliferate and continue to die throughout the brain. And because we used to talk about the blueprint for sex differentiation, the neuroarchitecture of sex differentiation. I don't use those words very much anymore because one of the surprising discoveries that we made along the way was, as I mentioned before, those steroid hormones, they are transcription factors, but they also appear to be able to modify the genome epigenetically. And epigenetic modifications are when every cell in your body has every single gene, right? But it only expresses a subset of it. So that's why a hepatocyte in your liver stays a hepatocyte and a retinal cell stays a retinal cell, right? Because if they started to express the wrong genes, you have to silence those genes epigenetically. But the brain appears to have a different set of rules when it comes to epigenetics. It uses it much more plastically to modify gene expression for, in response to experiences and things like that. And one of those experiences is whether or not you've been exposed to hormones. So a graduate student in the lab named Bridget Nugent was very interested in the epigenetic modifications induced by steroids during development. And she just looked at one of the modifications is DNA methylation. You put a methyl group onto a cytosine, the cytosine of, the guanine, cytosine, thymidine, and adenine. And she found that if she looked in the preoptic area, which controls male sex behavior, there was more methylation on average in females than males. So there was more gene suppression, right? And it turned out that's because in the males, in response to the testosterone, they actually turned off the enzyme that puts methyl groups on the DNA. So they expressed more cells, and the females silenced cells. Well, she can use a drug called zebulirin, which actually also inhibits that enzyme. the same way that testosterone does. This is actually a drug that's used in cancer therapeutics because a lot of cancers are escaped from epigenetic suppression. And if she gave that drug late, outside the sensitive period, it kind of peeled off the methylation groups on the DNA and it made the females sensitive again. So we came to the conclusion that the female brain has to constantly maintain a suppression of the male transcriptome in this particular region, because the male transcriptome sort of wants to burst through, and so she sort of maintains it in a silent state. But if you peel it back, you can get the male pattern. Fascinatingly, it turns out that's also something that's going on in the gonads. You think you couldn't get more terminal than an ovary versus a testis, right? But there's a particular gene in the ovary that if it is silenced at any time, if it's turned off, a testicular tissue will begin to develop. And you'll have what's called an ovatestes. You'll have a mix of ovary and testis. So we have to really rethink things about how permanent a lot of these changes are. It really opens up a whole lot about the plasticity that's potentially there. When I think about evolutionarily, what is the optimal way to go? You could be Mother Nature is like always hedging her bets. You never know when I might need to change back between the sexes, kind of thing. And so don't make it permanent. Just make it, enduring.
I see. Interesting. So, one of the things we've talked about the dimorphism at the level of the juvenile play behavior, and we've talked about it at the level of, what's going on in terms of what the cells are doing and things like endocannabinoid tone. So at least for the males, for juvenile rats, the males have higher endocannabinoid tone, and that is an important piece of why the play behavior differs between the sexes. We also know that cannabinoids and cannabis products in humans often affect men and women differently. And this brings us to the question of how dimorphic the endocannabinoid system itself tends to be in the brain and in the body. Can you speak a little bit about that?
Well, I don't know that I can. I don't think I have a lot of expertise on it throughout the body. I've really only studied it in the medial amygdala. So.
Okay. So one of the things I want to ask you too is it sounds like you've explicitly created rat models for studying the effects of cannabinoids, including THC, on development. So, rat models of, what is in utero THC exposure, what kind of effect is that going to have on the juveniles and subsequent adults that they develop into? What have you done there, and what results have come out in terms of what's already been published, and how does that start to make you think about the human implications?
Yeah. Okay. So there, we did a collaboration with Matt Hill at the University of Calgary, in which our laboratory injected pregnant animals with THC, and his laboratory set up to do THC inhalation through a vaping mechanism. And then Matt is the world's leading expert in measuring endocannabinoids and also THC in the brain. It's actually really, really hard to do because they're lipophilic molecules. But anyway, his lab measured the THC in the placenta and in the maternal circulation and in the fetal brain. And they measured also metabolites of THC, some of which are active and some of which aren't. Both the inhalation mechanism and the injection mechanism, because the argument being, well, people don't inject themselves with THC, right? They smoke it or they eat it. Both of those resulted in an enormous accumulation of endocannabinoids in the placenta. And in some cases, at some time points, an equilibration between maternal plasma, THC and metabolites level, and fetal brain levels. So an enormous amount was getting into the fetal brain. So that has implications, obviously, for cannabis use during human pregnancy. A woman named Yasmin Hurd at Mount Sinai has done a lot of work on humans and their ingestion of cannabis during pregnancy. It's increasing at a very rapid rate because of perceptions of safety. And I think we need to know, if it's safe or not. It might be safer than, say, alcohol or opiates, but we don't really know yet. And partly too is a lot of the literature that we had on it was done back in the 70s and 80s, and the THC content at marijuana at that time was completely different than, people were smoking a lot of oregano, right? And whereas now it's completely different. Yasmin has found Yasmin Hurd again at Mount Sinai has found some deleterious effects in humans, and she's tied that to some specific mechanisms. We so far, we don't see anything like, I would say, like deleterious per se. We see like natural variation in behavior, but again, it's just in our rats. But it really does speak to, I think, kind of like we know you shouldn't drink alcohol during pregnancy, and if you the question is why smoking cannabis during pregnancy. For some women, it's the nausea. And of course, nausea is very serious. It can be really devastating. But I guess if I were talking to a young person today, I would say don't think it's safe compared to alcohol, because we just really don't know yet.
Mm-hmm.
But it's definitely getting. It's like the old saying used to be, if you smoke, your baby smokes. It's the same for cannabis.
Yeah, and I suppose that even if we don't know a lot of the details yet, it sounds like it's safe to say that certainly cannabinoid exposure in utero is going to have some effect on the developmental trajectory of the brain.
Yeah. I think, we've done a deep dive into one particular brain region connected to one particular outcome. But the endocannabinoid system is one of the very first to develop in the brain, and the receptors for the endocannabinoids are considered the most abundant G-coupled protein receptors in the brain. So they're so ubiquitous and widespread that it's hard to imagine that there isn't some impact. And also that concentration in the placenta, and the endocannabinoid receptors are expressed all throughout the reproductive axis. So it might even not just be direct effects on the fetal brain. You start messing with the placenta, you're going to really impact fetal brain development as well. So, which has been shown in quite a few studies recently, some of them by Tracy Bale, who's shown that placental abnormalities following maternal stress can impact the developing brain. So yeah, we here in the States, we've been really hampered in our ability to study THC. It's extremely difficult to get. I have to have a DEA license. That's the Drug Enforcement Administration. It takes over a year to get the license, and it's tremendously tight monitoring of it. And, people always say, well, can't you go out on the corner and buy it, which is absolutely true. But I can't do research with what I can buy on the corner. I can buy it legally, through medicalization, et cetera. But because the reason I can get oxycodone or easier. I can get cocaine easier than I can get THC.
Wow.
And the reason is because there is no agreed upon medical use for THC. There's medical use for oxycodone and cocaine, right? And so that's one of the reasons there's such a push to legalize the medical use of cannabis, so that because we're really hampered in our ability to study it.
Mm-hmm. Yeah, we're getting a little bit of feedback from something on the desk, I think.
Oh, yeah, that's me messing. Sorry.
No worries. Another area I would love just like your general take on that. I don't know too much about this, but my understanding is, given everything that you study to do with development and sex hormones and all of this stuff, my understanding is that a lot of the stuff that we're humans today in the modern world, a lot of the stuff that we're eating that's coming in our diet, a lot of the stuff in our environment contains stuff, whether it's with trace amounts or even non-trace amounts of drugs and things that are influencing all sorts of stuff in our body, including sex hormones and things like this. So what can you say about that? Do you have any concern as a sort of neuroendocrinology person about the effect the modern human environment is actually having on our development and on sex hormones and all of this stuff?
Yeah. So you're talking about endocrine disrupting compounds, which are found in plastics and in fertilizers and fungicides and things like that. And it's such a difficult field. I used to do some endocannabinoid disrupting research, and the best evidence that they are in fact in the environment are having damage. It's not the dropping sperm count in men. That doesn't seem to be the case. It's the increased incidence of hypospadiasis in boys.
What is that?
That's when the urethra does not grow all the way to the tip of the penis, and it exits along the shaft. And it's it's a devastating condition, and it has to do with insufficient, impaired androgen action. It's very clear. We understand it very well, kind of thing. The other, and there's no doubt, if you measure the fat tissue, if you measure breast milk, etc., all of these compounds are in our bodies for sure. It's funny, back in my day, it was lead, right? It was lead in the gasoline and things like that. And we knew, definitely having effects, and got that all cleared out. You don't detect lead in kids' blood anymore, but now you can detect bisphenols and etc. And it's not as clean cut as the effects of lead were, but there's certainly the fact that we still allow RAID to be used, all of the pesticides and things like that there's big, it's scary. But that being said, humans are probably healthier now, Americans, other than our abysmal maternal fetal health statistics, but does not have to do with environmental plasticizers. The nutrition that we have now is so high that's I think those are counteracting forces. Puberty keeps advancing in humans, particularly in the Western world. I think, and particularly in girls, they estimate about 6 months a decade that puberty is occurring earlier and earlier. And it's really hard in that case to separate that out from the nutrients that we have now, the increased light, artificial light, etc., or endocrine disrupting compounds. What is sorting those out is so hard.
So puberty is advancing, you said, in human girls at about 6 months per decade. It's getting earlier and earlier. Interesting. What about in boys? Is that the same thing or something similar happening?
It's harder to measure puberty onset in boys. It seems more so in girls that they can do by breast development occurring. It's like they've moved down, like, because there's a thing called precocious puberty, right? So when you go through puberty way too soon, and it's as low as 8 years for African American girls.
Oh wow,
8 years old. That's considered precocious, but yeah.
And for some of these endocrine disruptors, could we just be explicit for people? What are some examples of these? And can you just sort of list again very clearly what types of consumer products they tend to be associated with?
Well, it's been a while. I'm not as expert on this as I once was, but the bisphenol A's was one of the big concerns, and they were at one point in a lot of plastics, including baby bottles and everything. And that's why now you look on the bottom of any plastic bottle, right, and it has that little triangle, and there's a number in the middle, and that tells you. But I think whether or not there's bisphenol A in there, I don't think any drinking bottles have them anymore. Then there are anti-fungicides that are used on like fruit trees and things. Fluozanol, I believe, that are anti-androgen like, and there's they're on grapes and apples, and like particularly like if you're getting grapes from Central America, which most of our grapes come from, Chile, and things like that. Those are a concern. There's the chemicals that we use, weed killers, raids and things like that are just, they're just bad. They're not just endocrine disruptors. They're just nervous system toxins. Anything that you're using to kill plants or animals is going to be killing something in your body as well. So
I see. So there's a lot of nasty stuff in the fertilizers and yard products that we use, in the food that we eat, and in the physical products in some cases, just plastic bottles and things like that.
Yes. And it's very, very hard. There's the number of new chemicals that are manufactured is crazy and hard to keep track of. And they can just change a single molecule. And again, just like how I said, how hard it is to study THC, it's really hard to study these compounds because the tradition is still sort of the classic toxicology, looking for a lethal dose, 50 kind of thing. And instead of looking for the more subtle reproductive effects and things. And then, there's all this evidence in things like amphibians and reptiles, and seeing lots of intersex, and, why are all the frogs dying, and et cetera. There's even things like birth control, steroid birth control is phenomenally stable. And so it can be detected in the water columns all the time, because just through the wastewater, because those compounds are designed to be stable so that they can pass through the human liver and not be digested.
I see. So you're saying that some of the molecules that come from the forms of birth control we use just are so stable they get out into the environment, and then we're all ingesting them at some level.
Yeah.
Interesting. And what are some of those compounds? What do they tend to be?
Ethylene, estradiol, norprogesterone, et cetera. Anything, that's in any of the classic birth controls.
Mm-hmm. Do you think this is related, plausibly at least? I don't know if we know the answer. Is it related to, the decrease in sperm counts that I know that we've been seeing dramatically?
Yeah, well, that's why I mentioned that. I don't think that those data are sufficiently reliable. The decrease in sperm count and the increase in breast cancer both suffer from the same problem of our methods of detection and quantification are so much better, right? So if you get better at detecting breast cancer, you're going to detect more breast cancers, right? And if you get better at quantifying sperm counts, you might be much more accurate. So it's been a while since I've looked at the literature on that, but I don't think I don't think that that's held up worldwide. There are certainly certain pockets where there are big effects on sperm counts and stuff, and particularly like, men who work a lot with fertilizers and things like that, or heavy metals. Heavy metals are always a big problem in industry and stuff. But this kind of global idea of has not held up.
I see. Well, we've covered a lot of ground already, a lot of fascinating stuff that you told us about. Is there anything you want to leave people with or summarize for people that we talked about, just to do with the general subject of sex differences in the brain and behavior?
Yeah, sometimes I get sort of pushback on that we shouldn't study sex differences in the brain because it can be abused, and that's always a risk with any science, and the idea that, it will just confirm existing biases about women's biological constraints. But I would say that that's not a reason not to, because of the discovery potential of studying sex difference. I don't just do it, like I said, for any. But my rats don't. They don't do mathematics. They don't write poetry. They don't. There's nothing to do with humans, cognitive abilities or anything in these studies. But what they do reveal is fundamental mechanisms by which the brain develops, and the enormous, beautiful, extraordinary mechanisms by which nature has found to develop the brain. And right now, the neuroscience textbooks are written based on the male brain. And they've missed a whole lot of varieties of ways that the problems have been solved in brain development. And that's, to me, is that's the reason I study it, is because it's like, I always have, I've got a contrast agent, right? I can see things that you couldn't see if you just studied one sex, because you wouldn't have a way of knowing it was different.
All right. Well, Dr. Margaret McCarthy, thank you for your time.
It's been my pleasure.